So with these collaborations, often extend beyond just products. So you already mentioned the llama. How has ACROBiosystems scientifically or technical support actually contribute to your success?
Absolutely. So I mostly developed natural killer cell therapies in my PhD. That's definitely my core area of expertise. But of course, because we can make any kind of immune cell in theory, we wanted to show in practice that we could actually differentiate to T cells as well as a bunch of other immune cell types. I've never done T cell differentiation before. I knew that the protocols were theoretically similar, but probably had a lot of differences. And I was able to reach out to the ACRO team and say, hey, as you guys have been developing this reagent, have you, gleaned any protocol insights or anywhere that you can give us a little bit of direction as we try these out? And they helped us out. They gave us some guidelines to test in a protocol, and the protocol ended up working great. And that's something that probably would have taken us weeks, if not months, of trial and error to get right, but because they had already been working on it, they were able to help us out, and that was like a really nice gesture of collaboration. I really appreciated that.
Yeah, I'm really glad to hear that. I was involved some of the early decision making for make that available to the public, especially customers. So I'm come to full circle that people actually reach out and actually use some of the guidance. That would be really great. Awesome. So from your perspective next, how have ACROBiosystems solution helped accelerate or de-risk your therapeutic development process? As you mentioned, a few things like beads to differentiate to NK cell.
Yeah, absolutely. So one of our biggest risks is tech transfer to a CDMO and making sure that we have a process that can actually work there rather than a process that works, in a well plate or flask, but is going to have to be entirely reinvented for commercial manufacturing. And so because we can use these beads and our process is truly scalable, once we optimize it in a 250 milliliter bioreactor or maybe a 2-liter bioreactor, a CDMO should be able to run that exact same process. There's no risk of having to reinvent or switch reagents or anything like that. And so knowing that that tech transfer can happen faster and at a lower risk profile is huge for our therapeutic timelines. Absolutely.
Definitely. I see too many things. Tech transfer from different scale of CDMO definitely is a topic that I've been encountered.
Terrifying.
Yeah, definitely.